So good day everyone. For today, I will be introducing the general characteristic or idea of Parastrongylous cantonensis. So the Parastrongylous cantonensis is commonly known as the rat langworm, and it is a causative agent of angiostrongyliasis, which is a disease that affects the brain and the spinal cord. Also, it is a parasite that has a characteristic of barber's pool appearance. So let us proceed to the introduction. Um, it was previously classified under the genus Angiostrongylus, and the previous name of Parastrongylus cantonensis is Angiostrongylus cantonensis. So what happened, or what are the changes that took place resulting in the changing also the nomenclature of this parasite? It is because in 1986, O.B. Laker actually split the Angiostrongylus cantonensis into 5 genera. So if the definitive host of the parasite is carnivore, then the genus must be called Angiostrongylus. And if the definitive hosts are murids, murids meaning rodents, then it must be called Parastrongylus. If the definitive host is mustelids, it should be called Angiocolus. And if the definitive host are gerbils and one murid, it must be called Galligostrongylus. So it must be if the definitive host is insectivores, then it must be called Stephan's Strongylus. So in history, it is first described by Shen Tao Chin in 1935 after examining Cantonese rat specimen. So yes, it is a rat langworm, but it was first examined on a Cantonese rat specimen. So it must be in China. Also, it is a nematode that causes angiostrongyliasis, and it is the common cause of eosinophilic meningitis. So now, note lang guys that there are a lot of causes of eosinophilic meningitis, but your Parastrongylus cantonensis is the most common cause. It is also common in Southeast Asia and Pacific Basin. So when you say eosinophilic meningitis, eosinophilic means there is an increased presence or population of white blood cells or WBC eosinophil. Then meningitis refers to the inflammation of your brain. So it is a meningitis with a high percent concentration of or population of eosinophils in your CSF or cerebrospinal fluid, and it is prominent in Southeast Asia and Pacific Basin. So also note that a that our Angiostrongylus cantonensis association with eosinophilic meningitis was identified by Nomura and Lim in 1945. So this is actually the first reported case of eosinophilic meningitis that is associated with the presence of your Parastrongylus cantonensis. So this is the geographical or epidemiological distribution of your Parastrongylus cantonensis, and this is the Pacific Basin. When you say Pacific Basin, it includes Pacific Rim, lands around rim or of the Pacific Ocean, and the islands of the Pacific Ocean. So, your Parastrongylus cantonensis is commonly resides in the pulmonary arteries of rats. That's why the common name is the rat lungworm. So these are the example of rats that has a worm in their lung organ. So snails are the primary intermediate host, while humans are the incidental host, also known as the ident or accidental host, where larvae develops until they are infectious. Humans get infected through ingestion of larvae in raw or undercooked snails or from contaminated water and vegetables. So, we have scientific classification of our Parastrongylus cantonensis. The kingdom is Animalia. The phylum is Nematoda. The class is Chromodoria. The order is Rhabditida. The family is Angiostrongyllidae. The genus is Parastrongylus. Then, the species is Cantonensis. So, we have the biology of our parasite. It has a bean-shaped bursa. In adult worm, it is a pale and a filiform filiform. When we say filiform, it is a thread-like. That's why it is called nematode, because it has a filiform or thread-like appearance. So it has a length of 17 to 25 mm. So it is it is quite small. So in male worms, it has a length of 16 to 22 mm. Then the diameter is 0.25 to 0.35 mm with well-developed caudal bursa or a kidney shape and single lobe. So these are the example of our worms in adult or in male worms. In the upper left side, as we can see the arrows, the VV stands for ventroventral. In VL, it is ventrolateral. And in LL, it is a laterolateral. In ML, it is mayolateral. The PL is posterolateral. The ED is externodorsal. And the dorsal race, which is the large arrow, has a scale bar of 50 UM. Male caudal bursa lateral view has a scale bar of 5 UM. So in the lower left side, as we can see the white arrow, it is a male caudal bursa showing gubernaculum lateral view that has a scale bar of 5 UM. Let us go back. So, also as we can see the lower right side of this image, as we can see the arrowheads of male caudal bursa of two spicules joined by a sheath, which is the arrow and has a scale bar of 100 UM. So let us proceed to the female worm. So it has a length of 19 to 33 mm. The diameter is 0.28 to 0.50 mm with uterine tubules that wound spirally around the intestine. So that's the reason why it gives a barber's pole pattern, and the barber's pole pattern can only be seen on an adult female worm. So the posterior end is blunt shape. Then it can lay up to 15,000 eggs daily. So this as we can see the image of the female worm, that is the example of the appearance of the Parastrongylous Cantonensis viewed under a bright field microscope. So let us proceed to the eggs. It is elongated and ovoidal with delicate hyaline shells. So it has a measurement of 46 to 48 UM times 68 to 74 UM. So it is also unembryonated when oviposited. So when we say oviposited, it is a zoological term for laying eggs. So let us proceed to the larva. We have different stages of our larva. So first stage larva, it can be found in the lungs of the rodents with a distinct small knob near the tip of the tail. So the two will develop chitinous rods below its buccal cavity is identified by the third stage larvae. So this slide shows the images of an adult female worm that has a characteristic barber's pole appearance. The first stage larvae and the second stage larva. So let us proceed to the life cycle. The life cycle of Parastrongylous Cantonensis is the adult worm of Parastrongylous Cantonensis live in the pulmonary arteries and right ventricle of the normal definitive host. So as we can see, the female lay eggs that hatch in the terminal branches of the pulmonary arteries, yielding first stage larvae. So the first stage larvae migrate to the pharynx, are swallowed and passed in the feces. So they are penetrate or are ingested by a gastropod intermediate host. After molts, the third stage larvae are produced, which are infected to mammalian hosts. So when the infected gastropod is ingested by the definitive host, the third stage larvae migrate to the brain where they develop into young adults. So if they develop into young adults, the young adults return to the venous system and then the pulmonary arteries where they become sexually mature. Of note, various animals act as parathenic or transport host. After ingesting the infected snails, they carry the third stage larvae, which can resume their development when the parathenic host is ingested by a definitive host. So humans can acquire the infection by eating raw or undercooked snails or slug infected with the parasite. So they may also acquire the infection by eating raw produce that contains a small snails or slug or part of one. So there is some question whether or not larvae can exit the infected gastropods in slime, which may be infected to humans if ingested. So for example, on produce. So the infection may also be acquired by ingestion of invertebrate parathenic host containing the stage 3 larvae, so which is freshwater or shrimp, then crabs. So in humans, larvae migrate to the brain or rarely to the lungs, where the worms ultimately die. So the larvae may develop the fourth or fifth stage in the human host, but seem to be incapable of the maturing or of maturing fully. So that is our life cycle of our Parastrongylus cantonensis. So for these slides, I will be presenting the different common names and the definitive hosts and the intermediate host. So for our definitive host, we have black rat, brown rat, and cotton rat. So for our intermediate host, we have slugs and snails. So there are the of our slugs and snails. So here are the pictures of our black rat, brown rat, and the cotton rat, as well as the different known intermediate hosts of our Parastrongylus cantonensis. So next, this is the another way of presenting the life cycle of our Parastrongylus cantonensis, but um, I will not be discussing this because I am done for our life cycle. So let us proceed to the pathogenesis. First, it is infected rats pass the worm in their droppings. Then the slugs and snails get the worm by eating rat droppings. Freshwater prawns, frogs, the crayfish, snails, and crabs get the worm by eating slugs or snails. So the people get sick accidentally by eating tiny slugs or snails on unrice. So yes, in the raw produce. So people can also get sick from eating undercooked slugs, snails, or freshwater prawns, frogs, crayfish, or crabs. So reminder, the first stage larva is the infective stage for the molluscan intermediate host, while the third stage larva is the diagnostic stage. So let us proceed to the clinical manifestation. So angiostrongyliasis, of course, usually associated with eosinophilic meningitis. So it is caused by the presence of larvae in the brain. So again, in the larvae, it is the third stage larvae and not the first stage larvae. So there will be the somatic symptoms. We have headache, fever, and malaise. But headache is the most common symptoms. Then we have the ocular angiostrongyliasis. So we have uveitis. The uveitis is an eye inflammation affecting the middle layer of tissue in the eye or uvea. Then blurred vision and loss of visual acuity or loss of visual sharpness. So let us proceed to the diagnosis. So eosinophilic meningitis of we have CSF projects abnormal chemistry. So it is elevated pressure, proteins, and leukocytes. and also eosinophilia. So it is rarely larvae can be found in the CSF or cerebrospinal fluid or brain tissue section. So this slide is the microscopic finding of a CSF sample under the microscope. It is a stained sample, so as we can see, there are a lot of eosinophils in the sample under the microscope. So we have also an example of those patients that has a presence of third stage larvae. So as we can see, the worms inside the of our positive patient of our Parastrongylous cantonensis, also in the eye. So let us also proceed to the molecular diagnosis. So real-time PCR assay, as we when we say PCR, it is for say specific detection of Parastrongylous cantonensis in human CSF. So it is offered by CDC. So it is also keep specimen cold with ice packs during transport, and the CSF volume should be at least 2 ml, because if the volumes is less than 1 ml, it may lead to reduced test sensitivity. So there are the treatment. So most patients recovered completely under about 2 weeks. So use of anthelmintics or albendazole and mebendazole. So it must be administered in combination with analgesics and steroids. So in our steroids we use prednisone. Yes, also the periodical removals of CSF can relieve symptoms of raised intracranial pressure. So this is the epidemiology or epidemiological distribution of our Parastrongylous cantonensis. So it is commonly in the area of Pacific Basin and Southeast Asia. So the infective area is the Australia, some area of Africa, the Caribbean, and Hawaii. So let us proceed to the prevention and control. So patient education must not to ingest raw or undercooked snails and slugs, potentially contaminated vegetables of vegetable juice. So removals of snails, slugs, and rats found near in the house and gardens. So thorough washing of hands and utensils after preparing raw snails or slugs. So thorough washing of vegetables. Um, that's all, and thank you for listening for my topic, the Parastrongylous cantonensis.